All right.
So, we are reviewing. There's no one in the middle. This is weird. We are reviewing the essay here. Transcription and translation. That's the essay that's on the next test.
Which is after Thanksgiving.
That's right. It sure is.
Yay.
But that doesn't mean you don't have to learn it now. Let's learn it now and then we'll know it. Because we got other stuff to learn after Thanksgiving. We got to learn the second essay. So, let's rewind this.
Oh my gosh, I'm so bad at this.
Rewind this. Okay. So here's the DNA that's in the nucleus. Everybody pay attention. Kobe, are you paying attention? Here's the DNA that's in the nucleus. And the first thing that happens is an enzyme. It's called RNA polymerase. It attaches to this promoter site. That's what the P stands for, promoter. Can you say promoter?
Promoter.
This is transcription. We're starting right here. And the promoter, this thing actually cannot attach to the promoter if there's a repressor protein in the way. The repressor protein is like an on-off switch. Right now, the gene is off. The RNA polymerase can't attach. Not happy. However, if this molecule is present, it could attach to the promoter, make the promoter change shape and fall off, and now transcription can occur. It's one way the cell can control whether the transcription occurs or not. It's called the operon system. And if you're taking honors, you have to read that section. Do you want to read that section who's taking honors? Did you read it?
Yeah.
Did you read it? See? Hey, no. The RNA polymerase attaches to the promoter site and slides down the DNA and opens up the DNA strand. And one by one will attach RNA nucleotides to the DNA chain. Click. Click. It's fine to click.
Wait, what are they
click.
clicking? What are the blue things?
These are RNA nucleotides. Click. You know the difference between RNA and DNA?
Yes, sir.
RNA.
Click.
They have different sugars.
Single
They do have different sugars. RNA has ribose, DNA has deoxyribose. What's another difference?
They're single-stranded.
RNA is single-stranded, DNA is double-stranded. Click. What's another difference?
RNA is in the nucleolus.
RNA is in the nucleolus, yeah. That's good. What else?
They're spelled differently.
RNA has uracil, DNA has thymine. RNA has uracil instead of thymine. You see what I'm saying? So, if this were TCAAGC, 10 points if you could tell me what the RNA will be that connects to it. 10 points.
Can we write the first letter?
Yes, you can just write the letters. You can just write the letters.
Oh yeah, the new has to go. Where does it go? Where the T.
10 points. Will it work? You know this thing right here, you can loosen that and then move that around and then tighten it back.
Yeah. Yeah.
And make sure you don't stop the video. Hey, it's a Kodak camera.
Peach, correct?
Yeah.
Meander highway through all the desks.
White correct.
Because like everyone in
Okay, I don't know if I know that. So.
Green correct.
Hey, did you dad find it, I think?
Purple correct.
Yeah.
Oh.
See, I'm telling you, it's like hard to get through here.
Oh, goodness.
Colby.
Alright.
Blue incorrect. Anyone else? Okay, so here it is. Listen if y'all missed it. Can y'all listen? Y'all with me? but it's not. It's U. Because RNA has U instead of T. A connects to U. G connects to C. C connects to G. You see? One of my favorite jokes. It's my favorite DNA joke.
What you see?
You see?
That's your favorite one?
It's one of my favorite ones.
What's your favorite?
I don't know. That might be my favorite. So, afterwards, we've made the string of RNA. What do you call it?
mRNA.
Shout out. mRNA. And the mRNA breaks off and floats away, and the DNA closes back up. And the RNA polymerase falls off at the terminator. The terminator site. And now, the mRNA is complete. And that process that I just showed you, that's transcription. Can you say transcription?
Transcription.
Now we're not done with the mRNA. We have to make some changes to it. We have to add a guanine cap. A modified guanine is put on the front, and we have to add a adenine tail. We have to add about 150 adenines on the end here to make this real long tail that is used. Scientists don't really understand it very well, but it's added onto the end. And then special enzymes called spliceosomes. They come up and they remove unnecessary pieces of the mRNA and they splice the ends back together. Splicing means to tie them together. And so now we have our final mRNA that's all ready to go. That part's called mRNA processing. What are you doing, a major close-up?
No.
It was on the thing that.
How are we doing? So 10 points, 5 points each. If you can tell me the 3 things that are done during mRNA processing. 5 points each. I just said them.
You got it.
Someone else want to write it while I hold this? Because
I'll write it.
you know it? Splice the stones.
Anna, did you not want to film anymore?
She was in the back and I kept getting in the way.
You could move up here.
I need to do it.
Or you could switch with Thomas, Anna.
You want to switch with Thomas, Anna?
These are not.
I would rather you switch with Thomas.
There is no order. They all have to
I
count
did all this right.
Why got all 3 right?
Wait, this is done at mRNA.
Processing.
Do you want me to hold it?
Why? Because it just is.
Purple, all 3 right.
Do you even know how to do it?
Oh.
Are you good at it?
Anyone else?
I'm writing it down.
Going.
Yeah.
Going.
The bottom one is not spelled right, but you know it. You know it. G.
2 right for green. Anyone else? No? I just showed this to you.
We know really.
Okay, so here it is again. For those of y'all who didn't get it. A guanine cap is put on one side. A polyadenine tail is put on the other side. Many adenines. And spliceosomes come up and remove pieces that aren't needed. and splice them back together.
Oh, it's not.
I didn't know what the tail was called.
We were talking about the one.
Mini adenine tail, guanine cap, splices them, removes pieces. I'm surprised that not everyone got that, so I just said it.
Just talked about, not like just like a little bit ago, where it connects to the promoter.
That's the RNA polymerase connects to the promoter and opens up the DNA and puts on nucleotides one by one to make this mRNA strand. That's transcription.
Oh.
Yeah, you should be a videotaper. You can video me now.
Now, what happens?
Then
Important.
Thank you, Isabel.
they make a protein.
It goes to
The ribosome.
the ribosome. I'm going to use this longer version here.
Oh, they get their amino acids.
Are y'all with me?
Yes, sir. The tRNA comes.
So, this is called what? This piece? Yell it out.
mRNA.
mRNA.
Is this translation?
This is called what?
Ribosome.
This is a ribosome. It's also called rRNA for ribosomal RNA. And what are these called?
tRNA.
tRNA.
tRNA for transfer RNA. The transfer RNAs carry amino acids. These are amino acids. That's transfer RNA. This is ribosomal RNA, and this is messenger RNA. There's a lot of RNAs to keep track of. I could envision a multiple choice question that said something like, what is this? And you have to answer if it's tRNA or mRNA or rRNA or xRNA. Yeah.
This translation.
This is translation about to start. We're going to translate the mRNA into a protein. Where's my little protein chains? Here's... No? I don't know where my protein chains went. Maybe they're on my desk back there.
Yeah, they are.
I'm sad. Can you throw me a protein chain?
Oh, I thought you said protein.
I did not remember that.
Pass, too.
My bad.
Okay, so this right here is going to be made into this. In this second part, it's called what?
Translation.
Translation. All at the same time?
Translation.
All right, here's how we do it. The tRNAs come into the ribosome. And this first three-letter sequence of bases, we call that a codon. The mRNA has codons. And these tRNAs have anticodons. This is the one everybody missed, the question that we had earlier. These things are called the anticodon. The anticodon on the tRNA tries to match up with the codon of the mRNA. And this one does not match up, so it floats away. And then another one will come in, and that one does match up. See how it matches up? So the first tRNA is in place. Hannah, are you getting any of this?
Yeah.
It seems as if you're not getting this.
Oh, I am.
Okay. Then the next tRNA will come in. Oh, I dropped it. And, oh no, I'm making a mess. Okay, the next tRNA comes in and tries to bind. That one doesn't fit. The next tRNA comes in and tries to bind. That one doesn't fit. The next tRNA comes in and tries to bind. That one does fit. And it binds right there. And now we have the next tRNA in place. And when the two tRNAs are in place in the ribosome side by side, a bond will form between the two amino acids. You know what kind of bond that's called?
Peptide bond.
A peptide bond forms. And then the ribosome slides down one codon, and the first tRNA leaves. And it will go get another amino acid, the same type it carried before. Now, does anyone know what this first codon in every protein is called?
Yes.
Starter.
Start codon. And do you know what the letters are for the start codon?
Yes.
AUG. And do you know what amino acid the start codon codes for?
Yes. Well,
Methionine. Methionine. This is called methionine. Meth for short. You always start with meth.
always.
Gets you going. It's a drug joke.
Guys, he's not serious.
Don't do drugs. But it's an easy way to remember that that's the first one. Okay, so the next codon is there, and another tRNA will come in and bind, and a peptide bond will form, and then the ribosome will slide down, and that tRNA will leave, and it will go get another amino acid, the same kind that it left there before. I don't happen to have the same kind, but just pretend.
Methylcycline.
What?
Methylcycline. What's the main name?
Methionine.
Methionine.
Methionine is the first one. Now the next codon is in place, and this one comes in and tries to fit. No, that doesn't fit. So another one will come in and try to fit. That does fit. That does fit, y'all. And a peptide bond will form, and the ribosome slides down, and this tRNA will leave and go to get another one just like the one it just dropped off. And this is just going to continue until we get to the last codon that clicks in place, and a bond forms, and the ribosome slides over, and the first tRNA leaves its amino acid there, goes and gets another, same kind that it dropped off before, and finally we come to the end. Actually, that happens about 300 times, because a protein has about 300 amino acids. Did somebody steal my protein?
4,000.
What happens to my... Oh, here it is.
Just losing everything up there.
Here we go. So, we actually have a whole bunch of these amino acids making a real long chain. But then the last codon codes for stop. It might be UGA. That's one of the stop codons.
Go dogs.
And the stop codon will come in and will bind. And this is the releasing factor is what we call this.
Go dogs.
And
Dogs.
the ribosome will slide over and kind of break apart there, and this tRNA will leave and go get another amino acid, just like the one it dropped off before, but look what we're left with. We're left with a protein. That's a protein right there. So we translated the mRNA code into a protein. Do you see what I'm saying? Know what I mean? Now, did y'all feel me on this? If a protein were 30 amino acids long, if there were 30 amino acids in the protein, 10 points if you could tell me how many nucleotides were in the mRNA. If there were 30 amino acids in a protein, how many nucleotides were in the mRNA? No, you got to write it down. A little math problem for you.
Professional.
Set it face down there.
Fine, Maddie.
White, 10 points.
Fine, Maddie.
Blue, incorrect.
You can't do that.
Green, 10 points.
I'm gonna watch this later.
Peach, 10 points.
You're gonna be like the Truman Show.
Orange, 10 points.
Right there.
Anyone else?
What's your favorite? Ashley, what's your favorite?
Purple, minus 10.
My favorite movie?
Okay, listen. Here's the thing. Nobody got it right.
The Conjuring.
Some of y'all got it sort of right, and some of y'all got it wrong. So let's pay attention here. Pay attention. If there's 30 amino acids in the chain, each amino acid requires 3 nucleotides to code for it. So, if there's 30 amino acids, you would need 90 nucleotides to code for it. But there's also 3 more for the stop factor, which doesn't carry an amino acid.
So it's 93.
So the perfect answer there would have been 93, which no one got.
Oh my god.
The next best answer would have been 90, which some of y'all got.
I thought it was 10.
Because there's 3 per each one.
Hey, bro.
Yes.
Can I get a sip of water quickly?
Yes. Okay? Now, the last part of the reading. So that's your essay. Any questions about this before we go on?
Yes.
Questions about this part?
I have a question.
Yes.
Is this chapter 22?
Yes.
Okay.
So there needs to be 93 tRNA. Wait, what are the little things?
This is mRNA down here.
That's my left hand.
This whole thing is mRNA.
But there needs to be 93.
That is an mRNA nucleotide.
Nucleotide.
So you would have to have 93 nucleotides if the protein had 30 amino acids. There's 3 per each plus the stop. Cohen, how you doing back there?
Good.
Are you on this?
I want to see K-12 reading it.
Cohen, that is awesome. Yeah.
Do we have a switch cam?
Yes, probably.
On chapters 21.
Heck yeah.
22.
Heck yeah.
22.
Heck yeah.
22. All right. The last part of chapter 22 talks about mutations. Have you ever heard of a mutation?
Yeah.
Let's talk about mutations. Hold on loosely.
Will you just get off my chin?
Hold on loosely.
Just kidding. I don't
Like, hello, 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 hello.
Singing hello,
I cheers.
Wait, why'd you say I look like?
I look good.
Because I don't do cheers.
I mean, really good.
I don't like you like.
Hey, everyone!
Oh my God, good.
Such a good
All right.
Oh my gosh, Shane. Oh my gosh.
Okay, let me tell you about how mutations happen. So, let's rewind. Let's rewind our minds.
Yeah.
Got it up here. Let's rewind our minds back to when DNA was being copied.
Wait, what movie was that?
Do you remember when helicase came in? Listen. Up here. Lachlan, up here. Remember when helicase came in and unwound it? And then DNA polymerase put new nucleotides on each side? Do y'all remember that?
Yeah.
And we made a copy over here and a copy over here. Remember that? And we ended up with 2 copies?
Yeah.
What if something went wrong when that was happening?
Oh my god.
What if this side was TCAAG, if it's DNA on the other side, it should be AGTTC, right? If it's DNA, if you're copying DNA, it should be AGTTC. But what if there was a mistake where the wrong nucleotide was put in? And instead of AGTTC, it was AATTC. That is a mutation. A mutation is when the wrong nucleotide gets put into the DNA strand during replication. And it can screw things up. How can it screw things up? If you have the wrong letter here, that could code for the wrong letter here. And could code for a different amino acid here. And that could change the protein. And so if you put the wrong amino acid into a protein, you could change how the protein folds. And you could change what the shape of the protein turns out to be. And the protein might not do its job right anymore. There's lots of cases of misfolded proteins causing problems. Have you ever heard of sickle cell anemia? We studied it last chapter. That's from a wrong amino acid in the protein that causes the hemoglobin to fold wrong, and that misshapens the whole blood cell. People with diabetes have misshapen insulin, and it doesn't do its job. People with dwarfism have misshapen proteins for the growth hormone, and it doesn't do its job. Almost every genetic disease is from a misshapen protein that was caused by a mutation when you're copying the DNA. And so here they show if the DNA is TTC, but it gets changed to TTT, that's a mutation because the DNA has changed. And if the TTC is transcribed to AAG, it will code for the lysine amino acid. So in other words, TTC codes for the RNA, AAG codes for the amino acid lysine. That's what they're showing here. Now some mutations are called silent mutations. And a silent mutation is what, like this last letter, if it changes from C to T, that'll code for AAA, but AAA still codes for lysine. So you get the same amino acid in the protein, and it doesn't change anything. It's a silent mutation. Shh, silent mutation. But if the TTC gets changed to ATC, in other words, you put the A where this T was, that codes for UAG, which is one of the stop codons. That'll stop your protein right in its tracks and make for a real short protein. We call that a nonsense mutation because the protein doesn't make sense anymore.
Is this still chapter 22?
Heck yes, chapter 22.
Oh my gosh.
In this case, listen. In this case, we've taken the TTC and we've put a C where the middle T was. So it's TCC. That will code for AGG in the RNA, which codes for the amino acid called arginine, which is a different amino acid than we had before. That could change your protein. All of these are examples of mutations. Let's see a video footage of a mutation.
Can we restart that from the beginning?
Mistakes, but it can happen.
Okay.
A point mutation.
Quality song.
Okay, pay
Everyone
attention.
makes mistakes, but it can happen. A point mutation changes a single base. This can change the whole amino acid, or depending on its position, could go unnoticed. Other mutations can be more damaging. A deletion removes a base, but also causes a frame shift, which changes every amino acid after the mutation. Originally, the codons would be formed in this pattern. But after the deletion, the amino acid sequence will change because the bases shift by 1. The resulting protein will likely be non-functional. An insertion adds a base, but also causes a frame shift, which changes every amino acid after the mutation. Originally, the codons would be formed in this pattern. But after the insertion, the amino acid sequence will change because the bases shift by 1. Again, the resulting protein will likely be non-functional. Lastly, an inversion will invert a segment of DNA end to end. Watch this section closely. Each base has been moved around. Again, these mutations have the potential to be harmful, but not all mutations have an effect on protein. Remember, during translation, a start codon must first be read to begin coding the protein. If the mutation happens before or after the protein coding sequence, there will be no effect on the protein. Mutations must occur in the coding section of DNA to affect protein synthesis. Otherwise, nothing happens. Thanks for watching this episode of Teacher's Pet. Don't forget to like and
Okay, let me say some things about that that are interesting. You could have a mutation. Listen.
Anna should never be able to videotape ever again.
I'm not there.
Listen. You could have a mutation affect every amino acid in the chain. And here's how that could happen. Let's think of the sequence like we would 3-letter words. I'm going to make a sentence with just 3-letter words. The cat ate
The mouse.
the
The bat.
rat.
Oh, 3-letter word.
The cat ate the rat. That makes sense, right?
Yeah.
What if there was a mutation where we just knocked out one letter, like the C?
The cat ate the rat.
Now, remember, DNA is read 3 letters at a time. So it's now D-ata,
Tetrat.
tet, her, at. Doesn't make any sense. That can happen in a DNA chain. We can knock out one nucleotide, and that changed the whole reading frame. It's called a frame shift mutation. And it changes every 3-letter sequence after that. So if we just knocked out one of these nucleotides, and we could change every amino acid after that. Every amino acid will be different. And you can have a drastic change to your protein. The protein can become very different with just one mutation. And usually that hurts the organism, but every once in a while it helps the organism. It makes them maybe a little bit faster, or maybe different color where they blend in or stand out or something like that, and it might help the organism. So that's the raw material. Mutation here is the raw material for evolution. Evolution is changing of organisms. Most of the time mutations are bad. It hurt the organism and make a bad protein that doesn't work. But every once in a while a mutation can make a good protein that's even better than what the organism had before. And so that's what we're going to learn about next. After we finish all this genetic stuff, we're going to go into evolution and see how organisms change. But make sure you read about mutations. That's the last section here. And that's what causes cancer. Cancer is caused by mutations. If you build up enough mutations in a cell, the cell could become cancerous. Could start reproducing out of control. And that has to do with what we already studied in cell reproduction.
They just happen.
One thing that can cause them is sunlight. Radiation can actually knock out nucleotides out of the DNA. Strong radiation from the sun, or radiation from nuclear waste, or stuff like that. Chemicals. Listen. Chemicals will knock out nucleotides. So you want to avoid smoking, drugs, all that stuff. That can mess up your DNA. And just aging. The older you get, the DNA starts to fall apart a little bit. So you usually see a few mutations every time a cell reproduces. And that can add up to big problems over time. Why is everyone getting up? I'm not done. Have a seat. Insane. Don't just start packing up before the class is over. It's exciting, I know you want to get out, but you got to let the teacher finish doing their thing. Okay? And that clock right there is a little fast, so we actually have a couple minutes still. So what do y'all think about this? Questions about mutations? No? Well, actually, I'll just go ahead and tell you, we're probably going to have a quiz tomorrow over 22, so make sure you read the section. Question?
There can be a mutation when one eye is one color and the other is a different.
That could be a mutation.
If they don't line up, the RNA, how would it work at all?
They don't line up.
Whatever Anna had.
Well, it could just make a protein that's a different color.
But the things would, you'd lose one and then none of them would line up, right?
I didn't know, but whatever you
Like the tRNA would not line up.
No, it reads 3 at a time.
Oh, so it's a
So if you
different-
lost one and those came together, it would be the next 3.
Oh,
The
okay.
next 3. You see what I'm saying? So it would still work.
The end. Say bye.
Bye.